Welcome back to Invodent embryology sessions. Have you ever wondered why our own siblings will not look exactly like us? To answer that question or to understand the phenomena, we need to really look into a process of cell division known as meiosis. So meiosis is a process of cell division which takes place in formation of gametes. During the formation of gametes, there is meiosis which is taking place to give rise to a haploid set of chromosomes. So to give rise to haploid set of chromosomes in the process of formation of gametes, this meiosis is taking place. So now, what are various stages in meiosis? It starts with a prophase. And the prophase is again divided into different stages in meiosis. The first stage in prophase is leptotene, followed by zygotene, that is followed by Pachytene, and that is followed by Diplotene. So these are various stages of prophase in meiosis. When it comes to the point of leptotene, in this, the two chromosomes are present. The chromosomes are clearly present, but the chromatids of the chromosomes are not distinguished. Can you see any chromatids? It is not. So, during the leptotene phase, the chromosomes will be present, but the chromatids are not distinguished. When it goes to zygotene phase, the chromosomes will come parallel to each other. This pairing up of the two chromosomes and coming close to each other is known as pairing. So this is the pairing. This pairing is seen in zygotene phase. And then what happens in the Pachytene phase? This you can call as a bivalent. You can see two sets. So it is bivalent. In this also the chromatids are not differentiated. But when it comes to the stage of Pachytene, we can appreciate that the chromatids are also present. So there are two chromatids which are present on the outer surface and two internal chromatids which are present. So now what happens is that there is coiling of two internal chromatids, and this stage is known as, this configuration of chromosomes is known as tetrad. This is bivalent and this is tetrad. Now the coiling up of the two chromatids which are present in the internal surface, which are causing the coiling, this coiling is known as crossing over. This coiling of the chromatids, is known as crossing over. The point at which the crossing over occurs, the point of adherence, is known as chiasma. Okay? At some point of crossing over, the chromatid gets adhered to each other. That point of adherence during the process of crossing over, is that point of adherence is known as chiasma. So the crossing overs and chiasma formations are all seen in Pachytene stage of meiosis. Then what happens in the Diplotene stage? In Diplotene stage, the chromosomes will try to separate from each other. When they are trying to separate from each other, there will be the break which will be formed at the site of chiasma, at the point of adherence. So there will be breakage of the point of chiasma by exchanging the genetic material, which can be appreciated in the metaphase. So in metaphase, we can see that the chromosomes are trying to split away from each other with a complete exchange of the genetic material between the two internal chromatids. When it comes to the Anaphase, there is complete split of the two chromosomes with the exchanged genetic material. If you can appreciate the change in the previous section of mitosis, where there is a longitudinal split between the centromere through the centromere, but here the two chromosomes individually are getting separated at the point of adherence, that is chiasma. So here the break point is the point of chiasma, whereas in mitosis the break point is centromere. So there is a split of the chromosomes which is taking place towards the centrioles and with the exchange of genetic material. And finally in the telophase, the two daughter nuclei are formed with two chromatids of each chromosomes, and there are two daughter nuclei with two chromosomes and each having the two chromatids with exchange of genetic material, which is not seen in mitosis. This exchange of genetic material is specially done only in meiosis. And then what happens? Till this section, this is all meiosis 1. And there is another phase of division after meiosis 1 is meiosis 2. So meiosis 2, again these two daughter cells are again divided each into other two cells. So each daughter nuclei is getting again divided into two daughter nuclei with a haploid set of chromosomes with exchange genetic material. So this is how the process of meiosis is happening in the formation of gametes. Hope you have enjoyed the session. Stay tuned to Invodent. Keep learning dentistry. Stay super motivated. Thank you.